So I've been asked to introduce our seminar speaker, Dan Barshis, today. Dan has an extensive and impressive resume, especially for someone as young as he is and as early in his career. Dan did his bachelor's at Evergreen State, and he then went on to complete his PhD at the University of Hawaii at Manoa in the zoology department, where he worked with Ruth Gates and Rob Tonin, studying the coral response to fluctuating environmental conditions. And then he went on to work as a postdoc at Hopkins Marine Station with Steve Palumbi, where he continued to study corals, stress response. After his postdoc, he jumped the invertebrate ship and went on to work with salmonids at the, as an assistant project scientist for the National Marine Fisheries Service. He just recently accepted a position at Old Dominion in Virginia as a faculty in what department?
Biology.
In the biology department, and he will be getting back onto the invertebrate boat after leaving on a little hiatus. Dan has a long list of publications ranging from the ISME Journal, PNAS, and MEPS. And I would just say, having worked with Dan, he is one of the most creative problem solvers I've ever met. And I think Dan kind of exemplifies why creativity is important in the scientific process in terms of experiment design and then just troubleshooting, problem solving an experiment you have going on. So without further ado, let's hear Dan describe his scientific creativity for us all.
Great. Well, thank you all for inviting me and hosting me. This is a lot of fun. I've been on that side of this building before, but never on this side. So I'm going to kind of walk you through a tour of the end of my PhD and my postdoc in the Palumbi lab. I'm not going to talk too much about NOAA fisheries today, also because officially I am not here on behalf of the NOAA Fisheries Service as my lab is shut down. And luckily I am a contractor with the University of California, so I am allowed to be here and still work today. But Tracy asked me particularly because of students here to sort of give you a quick tour of where I came from, and she described sort of the locations. I grew up in Chicago. I did my undergrad in Washington State at a very small state school, doing all things sort of field science and ecology, but they didn't really have anything marine science related except for salmonids. They're a little better now, but I kind of had to jump ship to the Marshall Islands, which I actually couldn't find on this map, but somewhere over here, as a volunteer with the College of the Marshall Islands. So I thought I wanted to study tropical marine science, and so I emailed people all over the world and found unlikely sap who took me on as a student volunteer in the Marshalls, and I did a few different trips out there, really sort of discovering my love for coral reefs as a system, and that's really what drove me to grad school in Hawaii. Even though I was going to school in Hawaii, I had to work in American Samoa, and then did a stint back in California in Monterey and Santa Cruz, and now I'm moving off the map to that uncharted waters of the other coast. But I sort of took a roundabout way to get where I am. I took these postdocs as a chance to explore the academic track until I decided not to, or until I was unable to, and I'm sort of continuing on that track until I decide not to. So there's a number of different options you can take with a career in marine sciences, and I'd be happy to talk to anybody about that afterwards. Oh, there's Virginia. I think. I don't know where Virginia is really. So my work kind of really begins at the level of the gene, but how those genes of an organism interact with different environmental conditions. So sort of a graphic here is the seawater temperature gradient along the west coast, and you can imagine an urchin in Victoria, British Columbia might be interacting with the same set of genes to a very different environment than that same species of urchin down in Baja California. And generally, the product of genes interacting with the environment is what we call phenotypes. These are different colors, sizes, and shapes, and the diversity that we see in nature. But I'm going to talk to you about some phenotypes that you can't always see and that you might not always classify as your sort of classical phenotypes, like metabolic performance or photosynthetic rate, or in particular, thermal stress tolerance. So all of my research sort of focuses on a few major research questions, the first being sort of what aspects of the environment matter. So what characteristics of the environment affect an organism's stress tolerance? And second, sort of on the mechanistic basis of which genes and physiological processes are really involved in that tolerance, that ability for an organism to interact with its environment. And lastly, sort of what the mechanics of stress tolerance can tell us about the resilience of an ecosystem or an organism. And I just modified this next part, but originally I described resilience as sort of very roughly as the ability to withstand change. Either an organism or an environment has some sort of optimal window of environmental change that it can withstand without shifting or crashing into an alternate stable state. When that environmental change gets too strong, that organism is pushed outside of the window, and this can lead to the collapse of a community or sort of the collapse of that particular organism's physiology. But in talking with my PhD advisor, and I'm going to a meeting next month, and he really sort of made me realize that it's worth getting into a little bit more. And so there's sort of two main types of resilience, or the ability to withstand change. The first being resistance. And that's sort of resisting your environment. I'm curious, who in this room does not know who this is? All right, I'm not as old as I thought. But, but here's a classic example of withstanding change. True to form, this is probably in the early 80s. In 2013, this is Mr. T. He hasn't really changed much, but it's sort of this ability to tolerate or resist a change in the environment. And the other side of resilience is really recovery, and that's sort of the ability to bounce back after an impact. And sort of the classic example with the coral reefs is a shift from this sort of nice, happy coral community with large predators and lots of hard coral cover to a more algal-dominated system. And this is what you talk about a phase shift, or sort of a lack of resilience in that whole community to absorb impacts without shifting to an alternative state. And so those are the two main sides of resilience. And the reason we're interested in resilience is, I'm sure as you've heard, the ocean is changing. There's this graph in carbon dioxide, and things are changing very rapidly. But we're already seeing the effects of these changes in the ocean in terms of changes in temperature, changes in pH. And we're really pushing a lot of organisms either at or approaching their physiological tolerance limits. So understanding how the genes of an organism allow it to survive these different rapidly changing conditions is sort of a timely question. And the other reason it's sort of an interesting time to be studying these types of questions is that the technology is changing. You guys have a very fancy machine down the lab, and we're now able to look at 20 to 200,000 genes in the same experiment. And these are just numbers to some of you, but I decided to crunch some numbers from my PhD. I generated about 720,000 bases of sequence data. Those are individual bases. This is a pretty modest data set. We were living in Hawaii. I had one technician who was also living in Hawaii, so this took us a few years. In my postdoc in the Columbia lab, I was working with 24 billion base pairs of sequence data. And so this is just to show you that really the scale of things have shifted over 5 orders of magnitude in the last, 4 to 5 to 6 years. And it's really revolutionized our ability to ask some of these questions and use these technological advances to gain a lot of insight in one big swoop. And now you can generate this much data in a few days. So it's really changed the landscape of things. My favorite landscape to work in is that of the coral reef. They're very pretty, they're very fun and exciting, they're highly diverse, they're very important. They occupy less than 1% of the world's ocean area, but host up to 25% of the marine fish, or fish species in general. They contribute a lot of protein to people dwelling near reefs and island nations. They provide a lot of goods and services, coastlines and tourism, and that's all sort of hand-wavy for people not like you guys. But you know, then they're also under a lot of threat, and they're sort of at the forefront of the debate over climate change and its impacts on ecosystem goods and services in the marine environment. There's a few options for corals in terms of what to do about climate change. You may have seen the sort of MAD framework before, but the coral could either move if the environment becomes suboptimal, it can adapt or acclimate, or it can die. There aren't that many options for a sessile coral, particularly something that's stuck to the bottom can only move by dispersal, not necessarily movement, or if tolerance limits get exceeded and they're not able to change, then they go extinct. Just to review, I'll focus in on that adaptation and acclimation, which is the sort of positive side of what corals are able to do. A brief review, phenotypic plasticity or acclimatization is when you exhibit a certain phenotype, in this case a blue phenotype, and the environment is nice and blue, and then the environment changes to a red condition and you change that phenotype. So the important thing in this case is that it acts at the individual level. It acts within a generation, so the same individual is exhibiting different phenotypes, and it's not heritable, it's not passed on. It does relate to your genes, but it relates to your ability to change the expression of your genes within the same generation. The alternative is to adapt, or sort of evolutionary adaptation. That's where you have blue and red individuals that are constrained in that phenotype. The environment changes, the blue individuals go away, and this happens across the population, across generations, and sort of passes on over evolutionary timescales. And so these are the two sort of processes that I examine in terms of corals and climate change types of stresses. And then the red genotype would then proliferate. Sort of the other base that we need to cover with corals is that they're not alone. They're in a primary symbiosis with these dinoflagellate algae called Symbiodinium. They also host a variety of other microbial partners, but we know the most about these. And this is important in terms of thermal tolerance because some types of these symbionts are more heat tolerant than others. There are different genetic groupings called clades, and in particular it's thought that this one clade confers greater thermal tolerance to certain corals than hosting a different clade. And this will come in to play during the second half of my talk, so we're just kind of bringing up this idea to start here. So we'll start with this first question of basically what in the environment matters in terms of the tolerance limits of a coral. And for that, I'll take you to our study site in Ofu in American Samoa. What sort of first got us interested in this study site was a paper by a National Park Service collaborator and my PhD advisor talking about these back reef pools. Basically, they were swimming around and it was very, very warm, and there was a lot of healthy looking coral. And you hear a lot about sort of coral thermal tolerance limits and bleaching and all these events, and they basically remarked, isn't that really cool? Let's measure it. So they measured the temperatures, they found that they were pretty extreme, and they measured the diversity of the system and found that it was pretty high. The way this works is, as you can see, there's sort of this back reef. It's kind of a moat system. It's dug out into these little pools that are circled here. And the reef crest sort of blocks the open ocean water from mixing in. So at low tide, when these back reef pools are isolated, the sun heats up the water. It's not mixed with cooler water, and you get this spike in temperature. When the tide comes up, the waves crash over the reef and mix, and so you get this sort of tidally driven cycle and environmental variability. So this created sort of a natural thermal laboratory. We have a site in front of the reef here that is constantly mixed, has more stable temperatures and smaller fluctuations. We have a medium variability site that's behind the reef, has sort of moderate temperatures and variability, and then a smaller water mass that gets even higher and shows higher degree of variability over the course of a tidal cycle. And so we can sort of compare corals in these different environments and see what aspect of the environment really matters in terms of the physiology of these corals. Just so you believe me, here's a temperature trace. We have blue is that fore-reef site, yellow is the back reef site, and red is the really high variability site. You see much higher maxes, daily maxes, as well as minimums, and just much larger degree of variability as you move into the more extreme areas of the back reef. And we've been working there for a while. It's not just temperature. Here's the maximum daily range and annual range in temperature, but flow, it's a flow-driven environment. Dissolved oxygen goes up and down. pH actually goes up and down. Steve's got a student sort of working on the pH aspect of this question now. But we've been able to quantify in the environment a lot of different things that change across this same scale. And so we've done a lot of field experiments, but what we were interested in was really honing in on what aspects of the external environment matter. And in this case, we asked the question of, do fluctuating temperatures actually change the thermal tolerance of a coral? So we isolated just temperature as a variable to examine its effect on the physiology of the coral that live in these different environments. So we did sort of a common garden acclimation experiment. We took 5 individual corals from the forereef, that more stable site, the more moderate site, and then the very high variability site. This is Parietes lobata. It's a mounding coral. All of the corals had the same genotype of symbiodinium, so in this case we don't have to worry necessarily about the effects of symbiodinium here. And we put them into 2 treatments, one sort of mimicking that forereef or more stable acclimation, and one mimicking the back reef and the more variable acclimation. We kept them there for about 36 days. This is in the lab of Jonathan Stillman up in Tiburon, California. So these are actually American Samoan corals grown in San Francisco Bay seawater, which might be one of the creative aspects Tracy was referring to. So we held them there in these different conditions, and then we examined their sort of terminal stress response, if you will. We really heated them up over consecutive days to push them to their extreme limits and look at how their responses differed based upon where they came from and sort of the conditions that they had been exposed to. And so the way you look at these types of data, this is just growth right after the acclimation. It's called a reaction norm, and I'll kind of walk you through how it works. You usually have your different environments. On the x-axis here, we have our stable treatment or our fluctuating acclimation, and then we have your response variable on the y-axis, and then you have your different origins within each environment and your different origins in the alternative environment. So what you look for is whether or not the different types of coral that came from the different locations are doing different things in the same environment, so here or here, just one half of the graph. And then you look at whether the same individuals are doing different things in different environments, sort of the same shapes in each half of the graph. And what I hope you can see here is that there's a difference between the different individuals here, and that there might be some influence of the treatment as well. And that's what we found. And so if you can think about the same genotype, in particular, these forereef corals grew more when they were exposed to a fluctuating temperature treatment. That's what we call phenotypic plasticity. It's the same genotype doing different things in different environments. But we also have a signature of adaptation, which is the different genotypes doing different things in the same environment. In this case, the corals from the more variable, high variability and medium variability locations grew more in both treatments, but particularly in that stable treatment. And so this is sort of a mixture of both adaptation and phenotypic plasticity, or a G by E interaction. So that was growth. The other thing we looked at as sort of a proxy for health is a parameter called photosynthetic efficiency. This is a fluorometric measure. You essentially measure the amount of light being used for photosynthesis. You expose the coral to a calibrated pulse of light, and you measure how much is fluoresced back. The sort of difference is what goes towards photosynthesis and heat and some other things. But it's a proxy for how well those algae are doing. And I didn't get into it, but the algae essentially contribute up to 100% of the coral's energy budget. And so if your algae are happy and you're getting a lot of nutrients, then the coral is happy. So it's a proxy. For the purposes of this, you just need to know that the higher the number, the more efficient the algae are, the happier we're assuming the coral is. So that's our response variable. We still have our different environments here. And this was the first day of our heat stress. So in this case, we saw different genotypes doing the same thing here, but a strong effect of the environment here, that the back reef and the forereef corals seem to do better in our variable treatment. There's a significant effect of acclimation. But we did this over 5 different days, so we pushed them to their limits a little bit each day. And so this was a result of phenotypic plasticity as well, but that some acclimation increases your photosynthetic efficiency. But if you push them across 5 different days, here's the temperature trace. Essentially, that graph is squished on top of itself for each day. We really cook them all the way up to a spike at about 40 degrees after 5 days, which is a lot for any coral to handle. But what you can see is that after, on about this 3rd and 4th day, that the circles really started to drop much quicker than the other shapes. And those were the corals from the four-reef. Regardless of their acclimation treatment, the four-reef corals, the ones that came from this stable environment, seem to be having a much harder time dealing with these temperature spikes than the corals from the back reef, or sort of native to those types of conditions. And this is a result of local adaptation, that the high variability and the medium variability corals seem to survive quite a bit longer than the corals that hadn't seen those conditions over a long time frame, not just the acclimation that we did in the lab. And so if we ask the question again, do fluctuating temperatures increase coral thermal tolerance? Well, they seem to under 2 different ways. There's some effect of acclimatization, where if you expose corals to a dynamic temperature regime, they're able to grow more and survive temperatures a little bit higher. But really the dominating effect that we saw was that corals native to those high variability environments seem to last a lot longer and have higher stress tolerances in the long run. And we know a lot more than that. This was my PhD, another Berkman student and one of Columbia's students, but we showed that, I just showed you that more variability in this case equals higher thermal tolerance. It also equals higher growth. We showed that these populations are in fact genetically differentiated at the coral host. So that was that Porites is actually different genotypes in these different locations. And then a different species, across a number of different species, we showed that clade D was more prevalent in our high variability habitat. So this is the Symbiodinium type that is associated with higher thermal tolerance. We find it more frequent in the more extreme areas of the back reef. And what I want to emphasize here is that, these are different sites, but this is really over the scale of 500 meters to about 4 kilometers. So it's really within what certainly a remote sensing type person would consider a single reef. And so we're seeing a lot of diversity both in sort of the genotypes, the physiology, and their ability to acclimate to different conditions across like a single reef tract on a small island. So now I'm going to move into sort of the more mechanistic basis. So we showed you that temperature variability matters, and it seems to show strong effects on coral growth and thermal tolerance. And so then we were really interested in what might be underlying those differences, what sort of cellular processes and mechanisms would be involved in stress tolerance. In particular, we can rephrase that question as sort of which genes are responding to stress, and are they different in a tolerant versus a more sensitive or susceptible coral? For that, we were relying on the work of Tom Oliver, one of Steve's students who was working with the Cropper hyacinthus. This is sort of your classic Pan-Pacific tabletop coral. It showed a similar pattern where the high variability corals had higher stress tolerances, but here there's another layer in that they were really dominated by that clade D symbiont. And we compared those to the medium variability corals, which had more lower stress tolerances, but a different symbiont type. So we have sort of the next layer of complexity in the coral symbiosis. And for that, we used one of these fancy machines. This was all the data I was talking about before. And it's pretty simple, but essentially what the technology leveraged now is that we're able to generate, tens of millions of sequences, but they're much shorter. So this is, 36 to about 100 base pairs. Now we're at about 150 or 200 or 300. But the smaller the sequence, the analogy that I use is if you heard, say, 1 second of a song versus 30 seconds of a song, it'd be a lot harder to identify the song if you only heard a second of it. And DNA is the same way. It's a lot harder to identify what genes these little snippets belong to when they're very short. So you can use a genome of your same species or something, but not everybody has a genome. So you kind of have to build your own genome, or in this case, a transcriptome, by using the reads and overlapping them little piece by piece by piece at very highly identical areas and sort of building that longer sequence. This is called a de novo assembly. And so you can actually build the different transcripts that come from your different genes in your genome. And that's the first step if you don't have a reference available. The next step is then to align the reads from each individual, say the purple individual. Now you can say all of these little snippets of sequence came from this gene, contig number 2. And there were 25 of them. If you remember basic biology, you have a gene that gets transcribed into a transcript. The number of transcripts determines how much protein you create. And so basically we just count the number of pieces of that gene and say this gene was expressed at a level of 25. This one was expressed at a level of 5. So there's a 5 times difference in expression. And that's sort of how these data are analyzed on a single piece-by-piece basis. But remember, there's up to 500 million of these little pieces, and so you actually need to learn to talk to a computer and tell them how to do this for you. And so here's the experimental design. We have a similar experiment as before, except with this coral. We took corals straight out of the environment. They weren't really acclimated to any laboratory conditions. We exposed them to a heated and a controlled stress treatment. Over 3 days, we sequenced the messenger RNA. We did our transcriptome assembly. We mapped the reads back to that reference. And then we asked simple differential expression analysis. Was there different genes being expressed in a different way between controls and heateds here, between controls and heateds there, or within controls between our 2 different types, or within heateds? So pretty simple design. And here's sort of how those data look. First, we had to build all of our references, and we came up with about 200,000 genes, or contigs. And there, just for sort of comparison purposes, there's about 30,000 genes in the human genome. So we don't think that a coral has 10 times as many genes as a human, but I did tell you that there's a lot of different organisms inside a coral. And so this incorporates a bunch of bacteria, a bunch of microbes, a bunch of ciliates, a bunch of that symbiodinium. So our first challenge was to figure out which were the genes that we wanted to look at. And so we kind of can compare them against really clean databases of coral sequence, and the genome came out while we were doing all this, actually after we had done all this, so we had to do it all over again. And take out some of the contamination, like the genes that match that symbiodinium, or ribosomal DNA, or other things that we didn't want to look at. And we came to about 33,000 coral genes. And we didn't expect to assemble all of them full length, but this is much closer to the predicted number from the genome of a related species. And about 70% of those we were able to put a name on. So the other side of this is that just because you have contig number 1 and it changes a lot, if you don't know what that gene, the protein that gene creates, it's kind of hard to figure out what it might be doing inside your coral. So we're able to match them to known proteins from other organisms and put a name on those genes so we can talk about things a little bit better. And so the first thing we wanted to do was just look at how many genes responded to temperature. So a simple question of how many genes changed their expression when we heated up the coral. So here is a graph of just those 103 genes in this is now our tolerant or high variability population, went up in expression, 56 genes went down. And the first thing you might see is that a fair number more genes seem to change in expression in the more susceptible corals. So you might imagine if you're more sensitive to a stress, you might react to that stress more, and that's in fact kind of what we saw. A lot of these were the same, though. The same genes responding no matter if you were tolerant of the stress or more sensitive to the stress. So we can think of this as sort of the core stress response. No matter where you've been exposed to or what type of coral you are, you might need to respond in these different pathways. But we were really interested in what was different. So first of all, there were more genes responding to stress here, and there were 81 over here that were unique to this comparison and 169 over here that were unique to that comparison. So we wanted to focus in on these and really look and see which kinds of genes were they and why were they not really responding in our more tolerant group of corals. So I'm going to jump right into a pretty complicated graph, but I'm hoping to walk you through this. I usually have another one in front of this, but I thought I'd try this out today. So each of these dots represents one of those genes, the 169 genes. Remember, these responded to the treatment in our susceptible coral, but were not significantly different in our tolerant coral. But we have the measurement. It just wasn't significantly changing between our treatments. So we can plot the ratio of those reactions to heat stress on this x-axis here. And what this means is that if the change in expression in our tolerant corals was greater than that in our susceptible corals, they're greater than 1. If the change in expression was less than our susceptible corals, there are less than 1 here. And you see most of the genes were not responding as much to stress in our tolerant corals. We're calling this like a lower reaction to heat stress. Means that the same gene didn't change its expression, changed expression much less than in the tolerant group than the susceptible group. You might think, or might be fairly intuitive, if something's not significant, it might just be because it's not that different from each other. But that's interesting. We have the same genes changing drastically in one group in a sensitive reaction to stress and not changing that much in the other group. On the y-axis here, we've plotted the ratio of control expression. So remember, we had a heated grouping and a control grouping. And so we can essentially use that control grouping as what are these genes doing normally. And we saw in this case, if the ratio is greater than 1, it means the control expression in our tolerant group was higher than the expression in our sensitive group. So they're starting off higher. So about half of them are in this upper panel here, and the other half are sort of just not changing as much in general. But as you don't change as much to heat in a tolerant coral, you often start a bit higher. So we're calling this sort of front-loading in gene expression. It means that these tolerant corals might be primed at a lot of the same pathways. They might be already active. They might be already expressing these genes and creating these proteins in a lot of these pathways that are necessary for stress, just normally under normal conditions. And this was sort of surprising. And you generally think of lots of gene expression as being costly, but in fact these corals seem to be expressing more genes, staying more tolerant, even growing faster in response to the environment around them. So something is definitely different at a number of these different genes. And so if we go back to the question of which genes are responding to stress, I didn't really show you a big table with a bunch of names, but if you trust me, there are many known pathways, stress response pathways, programmed cell death, or oxidative stress, or ubiquitin, or protein turnover. But not surprisingly, well, sort of surprisingly, once you look at the entire transcriptome, you see all different things changing. So there isn't really one core set of stress-responsive pathways. You actually see calcium homeostasis, components of different membranes, cell-cell adhesion, metabolism. You see a sort of across-the-board metabolic processes responding to that same stress and showing a different response between the tolerant and the susceptible groupings. And are they different in tolerant susceptible corals? Well, yeah, you really see this reduced reaction in the tolerant grouping and sort of constitutive front-loading in the stress-responsive genes. So these corals in the tolerant grouping really seem to be primed and ready to deal with additional stress. Steve Palumbi is really good at interesting analogies. They're interesting because they're not exactly what you would think of, but he was saying that, you know, a tolerant coral, it's like being in line at the security gate at the airport and already having your ID and your boarding pass out. So you're ready to go through the gate. I don't know how he got there from coral gene expression front-loading, but it's sort of one way to think about things. And so what I want to emphasize here is this was a lot of work of my postdoc and a lot of investment both in time and money, but just because we see this pattern, it's all correlational to this point. We see strong correlation between the expression profile of a given set of genes and the phenotype of interest in terms of thermal stress tolerance. And so really all this sequencing and all this new technology gets us to the point where we can start to ask questions of the right genes. Now we have, say, 169 genes instead of 30,000 that we might be able to target and say, okay, are any of these actually really crucial for the phenotype of interest? And that's important because you'll hear a lot about these new technologies, and it's just a new tool. You know, the questions are very similar, but it's a new tool to get us to the point a little bit faster of maybe being able to finally go in and see whether any of these genes are evolving differently, whether if you knock them out or mutate them, does that have an effect in terms of your physiology of your organism. And I want to kind of wrap up the other piece of the puzzle, which I haven't talked about until now. If you remember, corals also have this dinoflagellate symbiont that's very important for their survival. There's known correlations between heat tolerance and different genotypes of that symbiont. And in fact, in these corals, the more tolerant coral had a more tolerant type of symbiont. So we can ask the exact same question. Instead of whittling out all the symbiont and other genes to find the coral genes, we can whittle out all the coral and other genes to find the symbiont genes. And for the exact same experiment, the exact same data, just a different set of genes, we can ask, what is the symbiont doing? How are they responding in response to this heat stress? And we found a very different answer. And essentially, there was no consistent gene expression response to heat stress in the symbiont. Across 25,000 genes, either in our C clade or our D clade, we didn't see any significant response to our heat stress, whether or not you were a susceptible symbiont type or a tolerant symbiont type. And this was pretty surprising. I'll talk a little bit more about why this might be in a second. But the other thing we can do is do that within treatment comparison. Again, are C clade symbionts doing something different in the control treatment versus our D clade symbionts? And there we do see a lot of differences. We see out of about 4,800 genes that we could match to be the same in each genetic grouping, about 1,000 of them were showing a different expression profile, both between the control treatment and within the heated treatment, and almost half of those were the same genes showing the same difference. So fundamentally, these clades are doing very, very different things, but sort of in their steady state of existence, sort of within each treatment, not necessarily in response to the stress, which is sort of a different way of thinking about how these symbionts might be contributing to the physiology of the coral. And so if we put this all together to sort of ask what is the genomic picture of thermal tolerance, we have a constitutive front-loading or turning on of the different coral genes in response to stress. In particular, the more tolerant genotypes might be sort of pre-primed for dealing with subsequent stresses. And the symbionts were really unresponsive to the heat. And so we're sort of thinking about this and mulling this over now. There's a few different reasons that might have happened. One is that maybe, we did see coral bleaching, which is when you lose a lot of your symbionts, and so maybe all the stressed ones, all the ones that were reacting, were kicked out in the first few days, and after 3 days, we were looking at the ones that were still hanging on and not showing the stress. Alternatively, maybe symbionts inside a coral might be protected some way from the environment. They might be buffered from the external environment, and so maybe they're getting kicked out by the coral because it's responding to the stress, not necessarily because the symbionts themselves are responding to stress. And the last one, which will take a little bit longer to sort of figure out, is that looking across the broader literature in dinoflagellates, they seem to be doing things very strangely. They have really weird genomes. They have really weird photophysiological responses, much different from other plants. And maybe they're doing something very different at the gene expression level. And their proteins might be changing, but it might be happening after expression. So what we're seeing is not a change in expression, but we're essentially looking at the wrong stage in how proteins are created in the cell. And that's something I'm interested in following up with next year as I start up my own lab. And that sort of, these symbiodinium clades are actually very different depending at the same genes doing very different things in sort of a steady state profile. What I didn't really get into for time's sake is that, C and D clades have diverged up to 30, 40, 50 million years ago. So these are very, very different things. It's not actually all that surprising that of two different taxonomic groupings that are very distinct are actually functioning very differently. But it's going to allow us sort of some of the first insight into why they might be acting differently in a coral host. So what aspects of a D physiology might really influence the coral to be more heat tolerant. And indeed, we saw some sort of genes popping out in the components of the membranes of the chloroplast. And so it's known as you heat up a membrane, it gets much more unstable. And so a more heat tolerant symbiont might have different components of the membrane. It's also known that under normal conditions, the C clade, even though it's not as good under heat, it seems to be more productive and corals grow more under more benign conditions, and we saw some genes in sort of carbohydrate production and energy transport. And so that's sort of the nitty-gritty of everything, and I want to take a step back now and sort of talk about some of the broader implications of what can looking down a very, very specific path tell us about how these organisms are going to respond to change in the environment and how they're going to respond in the future. And so the implications of a lot of this research, I think I can confidently say at this point that Fluctuating environments seem to increase coral resistance in this case. They seem to increase the ability of a coral to withstand that change without a shift in the physiology or the community structure of those corals. Essentially, in this case, it's a sort of what doesn't kill you makes you stronger. If you're seeing a lot of thermal stress events that aren't going to kill you, they're going to exercise you enough to be able to deal with the next stress that comes down the line, much better than sort of a couch potato coral that's been sitting in a more benign environment for its entire life. So, you know, in terms of management and conservation, we can start to think about identifying some of these highly variable habitats where we do see healthy-looking coral reefs. And I could bet that it would be a safe assumption to think that those corals might be more physiologically able to deal with the changing environment than someone who hadn't seen that type of stress before. And sort of at the gene level, there isn't really a single resistance gene or gene family. We didn't find the gene that we could go sequence in corals and tell whether they're going to survive climate change or not, which isn't that surprising. But what was a little more surprising was that we sort of saw this sort of priming or front-loading of expression in the more tolerant corals. So maybe they're really just a lot more active. Something about them is allowing them to be much more active in the pathways that are necessary to respond to stress in the environment. And I want to come back to that sort of single reef scale. I've taken you through sort of growth and photophysiology and gene flow and genetic diversity all the way down to individual gene expression profiles. And we're seeing functional diversity from genes to physiology to adaptation and evolution really within this same habitat. Now, it's a pretty heterogeneous habitat. We have these different back reef pools. But again, it's along a very small spatial scale. So maybe there might be analogs to these different pools in other habitats where you might have a pocket of restricted water that gets a little warmer and variable than another area in a reef. I mean, we all know that reefs are very dynamic and different systems. And so thinking about the diversity that's out there, showing us sort of what potential these corals have to survive in the future, in my opinion, gives us sort of a little bit of optimism that basically everywhere we look, we see differences between these corals that have some direct relevance to how they can respond to the future of climate change. And so thinking about the winners of climate change coming from these more variable habitats might help prioritize some conservation efforts. And so I just want to leave you with sort of a picture of a coral reef and how the individual genes that a coral has at its disposal can be used to respond to the environment to create sort of the diversity of phenotypes, both visible and invisible, that we see out in nature. And why right now we have the technology to do this and perform these experiments across tens of thousands of genes. And it's really now in an era of sort of rapid change in the environment where learning and applying these technologies to really interesting organisms in the ecology and the environment that fascinates us is sort of a fun thing to do. And with that, I'd like to thank you guys for bringing me out here and a bunch of people who paid for and helped do a lot of this work. So thank you.
So my question is, do we know much about the genes that are involved in the bleaching response of corals, and whether those, can we identify whether those are changing in their expression response to these stuff?
Yeah, and I mean, like I sort of alluded to, is when you look at everything, you find a lot more things changing. And so I had a really hard time kind of, you know, you could take a heat shock protein or something that's known to respond in the bleaching response, and you'd find one that would be changing and three that wouldn't, and another one that might be. And so I think we're still, we're a bit far, but just because we can make a pocket genome in one study doesn't mean that it's as good as the genome of Drosophila or something like that. So I think we're still trying to figure out, but there is a lot of overlap, and there is building on some of the more model system literature in terms, particularly in yeasts and things, they find this sort of core suite of genes that change in response to almost every stress. And so they also find sort of the analogs of front loading in terms of more tolerant strains of yeast. And so that was pretty promising that at least conceptually there are these same patterns in more well-developed systems. But I mean, we're still looking at, there was a paper from one of Steve's other postdocs that showed a very strong bleaching response in the absence of any light. And so that just came out a week or two ago. And that if anything was really cool, but showed us that the model that we had been working with is that light creates a bunch of reactive oxygen species from the symbiont and from photosynthesis, and those go out into the coral cell, and everybody freaks out and it breaks down. And if you don't have photosynthesis, you don't have that ability, and you still have the bleaching response. So I think we're still figuring a lot out in that respect.
I had two questions. First, so methodological, and do you have any kind of independent validation of the gene expression data, qPCR, microarrays?
Here, no. In this case, we sort of validate by doing it again the same way. But yes, certainly that is the preferred way, would be to take a subset of these genes and compare their expression with a different technique. When people have done that, almost everybody that's published, which might reflect sort of the negative data type thing, but they find the same relative change in expression no matter how you measure it. It's just the absolute values of beginning and end seem to be a little bit different between techniques. But
RNA-seq is gaining acceptance in the
that was my experience, yes. And I well, no, I mean, it made it through review. But I would, when I said that sort of we're at the candidate next steps, personally, I would do a lot more validation before I picked a gene or two that I wanted to follow up with knockdowns and more in-depth analysis, because I believe in these patterns. Whether I believe that one particular gene is showing a consistent response, it's a little bit less, I would want to validate that quite a bit more. So I think in terms of what you wanted to do, I would defend the patterns that we see, but if you ask me whether this one gene was, I had a lot of confidence in exactly how it was responding, I would want to follow up with that.
So my conceptual question is, how do you think this would play out in population genetic sense in the landscape of the reef? Because clearly a lot of reefs are stressed, so they're not all resilient. So apparently being not resilient is adapted in a more stable fluctuation environment. And if that's most of the reef, then how is it that genes, I guess it's genes and the regulatory mechanisms can increase the frequency in these little patch areas? I guess really strong selection happening in those little patches, but then I have just in a population genetic sense, I have a hard time seeing how that's maintained.
Yeah, I mean, I guess the way that I think, and there's sort of an element, I alluded to it, but we have, back reef corals that are more stress tolerant. They grow faster no matter where you put them. They express a lot of genes, perhaps at higher levels under normal conditions, and yet they seem to outperform other corals across the board in however we've measured it. We haven't really gotten to fecundity and sort of reproductive output, which might be one thing, but classic theory would be that there's got to be some trade-off, otherwise everybody would be like that. My sort of hunch is that it might only matter in some of these pockets. So you have, even within a back reef pool, maybe right next to it, you have an area that's in a channel and gets a lot more consistent flow. And so they're dispersing into this pool, but as soon as you get that really hot, low tide, midday in the middle of the summer, all those guys get wiped out, and it's strong sort of post-settlement selection. It's there, so it's there in low frequency. It's just gets selected for increased frequency in these different habitats. I mean, you know, there's like the European eel example is like a good example of post-settlement selection. All the genes are everywhere, but then the environment sort of whittles away the ones that are not preferable, but they're always there. So it's not like a strong selective event of totally restricted gene flow and novel evolution. It's more maintaining the diversity of a population and then whittling away that diversity in terms of where it's a more extreme environment. So I kind of see it as all the genes are floating around, but they're sort of preferentially selected for in some of these environments. And we do see, at least in say, ITS and mitochondrial markers, we see some leaning towards reduced diversity in these more extreme habitats compared to the rest of the population. And so that kind of fits that model. I'd like to test that and look at, you know, I put out some settlement tiles to see who's really making it between these different environments, but I haven't had the chance to go forward with that.
It's true that the genes are everywhere, and maybe these catchers are not necessarily a focus for consideration.
I think so. It depends, though, how much, you know, if there's sort of a double layer of acclimation and adaptation, you know, if you're already acclimated to deal with stress at higher stress tolerances as well as adapted to as well, then maybe if you're not acclimated to, you might not be able to survive. But I don't know. It's.
I can see the advantage of overcoming the Lee effects after some major advantage.
Yeah, and that'd be the other thing, is that there's just a lot more reproductive material to sort of recruit and settle to neighboring areas after a big die-off event. I mean, we've been working in a national park for 8 or 9 years now, and you know, in the last 10 years, they haven't had a huge bleaching event, and so we're sort of looking at things in a somewhat unique environment. I mean, I say this in sort of how this research applies to other environments without having gone out and looked, and so I'd love to go somewhere else and see, do these same patterns play out. There's some people in the Persian Gulf that see more stress-tolerant corals coming from the sides of the reef that do get hotter and don't get as much circulation. So it'd be like the leeward side versus the windward side of a reef. And they have almost the exact same sort of temperature traces between these different environments, and see some differences. What I think really matters is at the sort of individual coral genotype to individual symbiont genotype level. And there's Ileana Balms is working in the Caribbean, where there's much less diversity in symbionts and corals, and so she's able to get down to the same clone of coral within the same C or as an A type of symbiont. They can actually find different genotype by genotype combinations that have pretty different stress tolerances. So I think a lot of this work is sort of looking at things at a much bigger level, but we're starting to see every individual coral is kind of doing things differently, and now by trying to get to that individual genotype scale might allow us to answer a few more things. But great, thank you.